Hi guys, today we're talking about enzymes, and enzymes are important molecules to our metabolism. So let's start with a quick review of what metabolism is. All of the chemical reactions that go on in your body, we collectively kind of call metabolism, and there are really two categories of metabolic reactions. Anabolic reactions are also called synthesis reactions, so like photosynthesis, where you're working to create molecules. And photosynthesis is just one example. There are lots and lots of examples of molecules that you create inside of your body, and that involve synthesis reactions. And then also you have reactions which we call catabolic, which take apart molecules. Sometimes we call those digestive reactions as well. Cellular respiration is largely a catabolic process where you're breaking apart a sugar molecule. And when you do that, what you're trying to do is get ATP. ATP consists of an adenosine unit that's connected to 3 phosphate groups, and I know this is not the chemical formulas that you're looking at. It just kind of is a quick example. But this is the basic energy molecule that cells use, and the energy is in the bond that holds the last phosphate onto this ATP molecule. And that bond is worth 7.6 kilocalories of chemical bond energy. Some of you I know are thinking right now, what about glucose? I thought glucose was the energy molecule. And you're not wrong. Glucose is the big energy molecule. The problem with glucose is that it contains about 686 kilocalories of energy, which is way too much energy to power most of the cellular processes that your body does. So anything that requires energy, on the cellular level, might require up to maybe 15 kilocalories of energy. So if you burned an entire sugar molecule to do that reaction, you would have the rest of the 686 kilocalories wasted just to do that 15 kilocalorie job. So instead, through cellular respiration, glucose is broken down to create this ATP molecule. And because ATP has a smaller energy input into creating this bond, there's less waste when you use that molecule. All right, so let's talk about enzymes as catalysts. Enzymes are proteins, right, most of the time, and they act as catalysts. Remember that a catalyst in your chemistry class was something that generally helped the reaction to go more quickly. Essentially, they change the rate of the reaction. Also in chemistry, remember you have the arrow between the reactants and products in a chemical reaction. Typically, the catalyst would go above that arrow right there. The reason why it goes in the arrow is it's neither a reactant nor a product, but it is required to make the reaction happen. And so it's not consumed by the reaction the way that the products are, so you don't put it down into the equation. Enzymes' primary job as catalysts is to lower activation energy. And remember, activation energy is the energy that's required to take reactants and get them to a point where they will start to react to form products down here. And you can't see the word product, but that is product right there. So if you look, this black arrow right here is showing you that from here to here, where we've started with the reactants at this dotted line, that's their energy to begin with, we have to raise their energy to this point before they start to convert to the products. When you add an enzyme, notice you don't have to go as far. The magnitude of this arrow is much smaller, and therefore you don't have to raise the energy that very much to get the reaction to go. Now, when activation energy is required, usually it's in the form of heat, and that, again, from chemistry ought to make sense, since you guys usually cooked a reaction, you put it over a Bunsen burner or something like that when you wanted to get it started. The idea there is that you raise the kinetic energy of the molecules to increase the number of collisions between them to try to get them to react with one another. All right, more about enzymes is that they're specific. Remember, they're made of proteins, which means all of a sudden shape becomes important, right? Shape is important to the activity of most proteins. Enzymes bind to the reactants, which we call substrates here, because they match the shape. So the substrate's shape matches the shape of the active site of the enzyme. So there's a picture here at the bottom, and you can't see all of it. It kind of gets cut off down here. But here are substrates, and notice that they fit perfectly into this active site of the enzyme. Now, what we didn't discuss in baby bio that becomes important here is this idea of induced fit. If you actually look at the enzyme before the substrates bind to it, notice you've got a lot of space in here. There's a lot of room, and when the substrates actually come into the enzyme, and you form this enzyme substrate complex, notice there's a lot less space in the active site of the enzyme. Essentially what happens with induced fit is it's kind of like the enzyme hugs the substrates. It changes its shape ever so slightly to kind of conform to the substrates. You can think of it, some people say lock and key, which I guess makes sense if you understand how an internal lock mechanism works. But I think the easiest way of thinking of it is like a handshake. Your hand has a specific shape when you go in to shake somebody's hand. And then your hand conforms to the other person's hand when you lock hands to do that handshake. It's sort of like that. So here's the substrate coming into the enzyme, and then what you're seeing here, and it's a little difficult, it gets cut off, but notice the color change. This we now call product, and it gets released, right? And then notice once the product comes off, the enzyme goes back to its original conformation. And because it does that, the enzyme is therefore reusable, right? We're able to bind more substrates into this active site. It's there, it's open. Therefore, you can keep using enzymes over and over again to carry out chemical reactions. Now, there are things that can actually happen that can change the activity of an enzyme, and the one that you learned in baby bio was denaturing, right? Where you have an enzyme and it's got this specific shape, right? And it's supposed to fit a specific substrate, right? But you change the pH of the solution that the enzyme's in, or you change the temperature. Usually it's low pH and high temperature, but also high salinity. High salinity can interfere with the tertiary and quaternary structure of the enzyme and make it kind of fall apart. So now what you end up with is an enzyme that has an active site that might look something like this, and now it doesn't fit this substrate anymore. So pH, temperature, high salinity can change the enzyme shape. Also, you have cofactors or coenzymes that can actually help to control an enzyme's activity. So oftentimes these can scaffold, so they can kind of hold the active site of the enzyme into a particular shape so that it can still be active outside of maybe, say, a normal temperature or pH range. Those cofactors or coenzymes are not made of protein. An example of this is some of the vitamins that you take. They kind of bind in here and scaffold and hold the active site into a specific shape. In addition, in terms of controlling enzymes, you also get something called inhibition. And inhibition means that you are slowing the ability of the enzyme to cause the reaction. And there are actually two types of inhibition. The first of these we call competitive inhibition. So a competitive inhibitor is a mimic. It actually can fit the active site of the enzyme well enough that it can block the active site. And I actually have a non-competitive inhibitors bind to another site on the enzyme, so they don't actually bind into the active site, so they're not competing for access to the active site, which is why they are non-competitive. But what they do when they bind to the enzyme is cause the active site to change, and a lot of toxins and poisons will do. This is a form of something that we call allosteric inhibition. Allosteric means other site, basically, if you look for enzymes. So this is similar to non-competitive inhibition, but when you're talking about non-competitive inhibition, this is something that can permanently change the shape of an enzyme. Allosteric regulation can be allosteric inhibition, but it can also be allosteric activation. So say, for example, you have an enzyme that's particularly active and you're concerned that it's going to damage a cell. You can do one of two things with that enzyme. One thing you can do is manufacture it to be misshapen on purpose, but then when this non-permanent regulator molecule binds to the enzyme, it actually stabilizes the protein shape, causing the enzyme to become active. So now you've got a digestive enzyme that could digest the inside of the cell if you needed it to, but you can turn it off by removing that activator molecule. So you can actually destabilize the enzyme by removing this allosteric molecule. So here's another place on the enzyme and causing it to change shape, someplace other than the active site. It's an allosteric inhibitor or activator. There's also something called cooperativity, where enzymes have multiple active sites. So once you have binding at one site, once you have induced fit at one active site, then all the other active sites on the enzyme get stable of that here. So here's what the enzyme looks like normally, right here in this diagram. The substrate here should fit into the active site of this enzyme. But when you have a competitive inhibitor in this next picture, notice it doesn't fit exactly, but it fits well enough that it can still block the active site. Now, this substrate can't bind, and because it can't bind, this renders the enzyme basically useless. And that's what you're seeing in this diagram here. Notice here is the site where this non-competitive inhibitor is binding, and when it does, it kind of tugs the edge of this enzyme that way, which means now the active site of this enzyme doesn't even function properly, and you don't get binding to the enzyme by the substrate. Diagrams here, right here, right here, and right here, this is the allosteric site. It's a binding site for something else, which can help us to kind of act like an on-off sample of that. Okay, so this is our enzyme. It doesn't take its proper shape until the activator is added to it right here. So now we've got this stabilized active form. If you take this off and replace it with an inhibitor right here, notice it changes the enzyme to the inactive form, and it just kind of stabilizes it that way. So removing the activator can actually destabilize the enzyme, and then the inhibitor, when it binds, will actually stabilize that in its inactive form as well. So anytime you have something binding, so here's an example of that right down here. Okay, so we've got this inactive enzyme, we bind a substrate to it, and all of a sudden now every other active site is the proper shape so that that enzyme can act, and that's called cooperativity. It's really cooperativity between the active sites. Also, we have metabolic pathways. Where enzymes are concerned, often they're involved in multi-step reactions, and those we call pathways, right? Chemical pathways, you might be familiar with these in chemistry. So these are reactions that occur in series where the product of one step leads to the binding of a different enzyme to create the next product which does the same thing. So the biochemical pathway that you see here is going to take threonine, which is an amino acid, and convert it to a different amino acid, isoleucine. So when there isn't isoleucine present in the cell, threonine will bind to isoleucine to create intermediate A. this will bind to enzyme 2 to create intermediate B, which is now able to bind to enzyme 3, and so on. And that's what you can see. You create these intermediates, they bind to a different enzyme that's waiting to bind to them until you get down to the end, and here's our end product. That's what we wanted. Once this goes on long enough and you get a bunch of this isoleucine, we don't need a bunch of isoleucine hanging around. And so that isoleucine is free to go and bind to, lo and behold, an allosteric site. This shuts off the enzyme because notice the active site changes shape. Now this enzyme can no longer bind to threonine. When isoleucine runs out in the cell, then this isoleucine molecule that's attached will fall off, which is what you're seeing here, which takes our enzyme and converts it back to its active form so that the biochemical pathway can begin all over again. When you have the end product of the reaction shutting off the reaction, we call that negative feedback or feedback inhibition, because the end product is stopping the reaction from moving on. Finally, there's one other concept I just want to kind of sketch out for you here, and that is the trajectory of enzyme-catalyzed reactions. So if you have the rate of the reaction here, and you have time on your y-axis here, often enzyme-catalyzed reactions will take a curved path like this, and that's not the prettiest of graphs, I know, but essentially you have these three main stages, right, where you've got this slowed reaction rate here, you've got generally equilibrium at the top, and then you have this slow reaction rate here. What's happening here in this part of the graph is the enzyme is just getting started, right? And it's binding to substrates, releasing them as products, binding to new substrates, releasing them as products. Enzymes get to a point where they will reach their maximum speed at equilibrium, and that's what you're seeing here. Okay, so initially you've got lots of substrates here in the first part of the graph for the enzyme to bind to. And so the enzyme will really get going. It takes a minute for it to get up to its maximum speed, but then it reaches its maximum speed. And the enzymes can only go so fast, and that's the reason for this equilibrium portion. But then something interesting happens. You start to get to a point where your products have reached critical mass. So here you've got more reactants. Here you've got more products. So what's happening is the enzyme is encountering less and less substrate as time goes on. The concentration of the substrate has dropped to the point that it's getting difficult for an enzyme to find substrate, and so the reaction rate starts to dip, like you see here. So something else that can influence the rate of chemical reactions or enzyme-catalyzed reactions is concentration. If you wanted this reaction to go faster, you could always dump more enzyme into it. Adding enzyme here helps to boost the concentration of enzymes, and as those enzymes reach their maximum, you would see the rate of the reaction go up as well. So that's all I can think to tell you about enzymes at this point. We will be meeting in Google Classroom and talking about them very soon.